Another announcement to make. We told you in a letter that on Monday, since this is the summer school, but not only for lecturing, but also for exchanging, kind of networking and exchanging information, we would like to have, as we traditionally have, to have a poster presentation of your work. So we're going to have a poster session on Monday afternoon, I believe, where we expect that you present your posters, some interesting research that you're doing. It doesn't have to be nothing, big, at least some methodology that you're using and some data that you have. So if you brought your posters, great. If you didn't bring your posters, we would appreciate if we get them on the memory stick, or we can collect them as a PowerPoint slide so we can print them here. The sooner we start to collect them and print them, the better, because I don't know how many of you need to print the poster here. Okay, it's not too bad. But I think we should get started Wednesday or Thursday. Mondays, is a holiday. So I don't want to give those people in IT all the posters on Friday to print. Table and Fred are going to collect those PowerPoint slides during the week, and then we're going to print them out and we're going to have the poster session. They promised some awards. I don't know. We'll see. Okay, so as I told you, my name is Marina Marjanovic. I'm actually Associate Director of Imaging in Illinois. I work with Professor Steve Boppard. And I'm also Adjunct Associate Professor in the Department of Bioengineering. But I have a completely different background, so we're going to switch gears a little bit. And I apologize for my voice. We had a big commencement in this past weekend, so I was sitting outside in the stadium for quite a few hours, so kind of got a little cold. I'm actually a physiologist, and but I'm involved in imaging and different types of imaging. But I was doing my graduate work here at U of I in physiology, but as a postdoc, then I actually did physiological NMR. So I kind of switched gears a little bit and started working in NMR field. And simply, biophotonics, as the name says, it's not only about how you image, but what you image. And very often your target is some biological sample or even some translational study, right, in a clinical setting. So in order to really successfully interpret your data, you not only need to know your modalities, but you need to know your target, your samples that are, you probably know, very complex. So how many of you use any biological samples or biological targets? Quite a few of you. And you need to know their nature and their characteristics so you know what you're looking, right? And specifically, a lot of you are not only looking at the tissues and cells, but also we are very much interested in certain pathological conditions. Can we recognize them? Can we differentiate them? Can we treat them, right? So I'm going to take you back to your high school and give you a very fast overview of what's important to know about the cell and tissue. And then we're going to actually work on one of the probably the most important targets when you talk about biophotonics, and that's cancer. Because I really don't know how much do you know what is the cancer and how the cancer actually develops, so you can recognize it better in using your imaging modalities. So first of all, you know that there are different organizational levels of life. We are all made of what? Cells, right? The cells are organized into tissues. Tissues are organized into the organ systems, and organ systems are organized into a coordinated organism. Of course, cells are very complex units, and the more we develop different modalities, the more we know how complex these structures are and how well they're coordinated and synchronized. They're made, of course, out of the organelles. Organelles are made of macromolecules, and macromolecules out of the simple molecules. But it's very hard to define what is life. But we know one thing for sure, that the smallest unit of life is a cell. Cell is the smallest unit that has all the properties that we define as life. If you take, you know that if you take any organelle or the molecule out of the cell, it's not life. It's not alive. So you have to be very careful about that. And of course, even if you take the cells out of the organism, you know that you're losing a lot of properties that the cells have as a part of the tissue or the organ they're in. So the goal of our imaging efforts are always to try to do as much as possible in vivo. So cells, as we mentioned, are a very complex unit. They have cell membrane, they have different organelles, nucleus, mitochondria, endoplasmic reticulum. But do you know what is the most important organ that actually defines the cell, without which the cell does not exist as a entity? No, that's not the organ. Membrane. Membrane is the organelle without which there is no a cell. There is that main barrier that actually defines what's inside from the outside of the cell. And very often, you know, people just ignore the cell membrane and say, well, it's not. Well, it is an organelle, and it's a very important organelle. It has all the characteristics of the organelle. So, and it regulates the passage of course, materials between the cell and its surroundings. And we have a DNA, but again, we have also cells where we have RNA instead of the DNA, right? Yes?
So you said membrane has all the characteristics of an organelle. So
yes.
How do you define organelle?
It has a specific, it's a part of the cell that has a very specific structure and function. So it has you know that it has a very specific structure and the function, right? It has its certain functions so we don't go like regulates the transport. It has receptors to receive the signals and transport the signals inside the cell and so on. So the main parts of the eukaryotic cell, of course, is the membrane, the cytoplasm, and the nucleus, and of course numerous organelles that are within the cytoplasm. And without going into any details, this is the very simple representation of the cell membrane. It has the lipid bilayer. It has a lot of membrane proteins that are either sticking on one side or transmembrane proteins that are going from one side of the membrane to another. There are very important cholesterol molecules that are inserted within the membrane. And also you can see that not only that the membrane itself is very complex, but the extracellular matrix that's outside the cell is also having a very specific structure. And I'm mentioning that because when we start now talking about the cancer, we'll see that now more and more our knowledge of ECM or extracellular matrix is very important, because it's not just the fluid or a solution outside the cells, but it has a very defined structure and very important function, or how the cells communicate with each other and how they develop. So let's talk about one important property of cell, and that is cell reproduction. Cells divide, right? Cells reproduce. Why do they divide? Well, for three different reasons. First of all, in the case of unicellular organisms, the division of cell brings, new organism or the new cell. So it's simply a reproduction, like in the case of viruses or bacteria. Of course, the cell division is very important in the growth and development. We all start from one cell, and cells divide, and we grow, and then they differentiate and specialize, and that's how we develop. Now, even if when we reach a certain growth and development, certain cells still need to divide. Why? Because many of them get injured, get mechanically or chemically injured and need to be replaced. So there is always importance of tissue renewal. And certain cells in our body have much higher capacity to divide, and others lose that during our development. For example, cells that keep the very high potential to divide are the epithelial cells. What are the epithelial cells or epithelial tissues? Those are the tissues that line up all your cavities and the surfaces, not only of the body, the skin, but also the surfaces of the organs. And those cells are the first and the most important barrier between your organs or your surface of the body and the outside environment. And of course, they are first to be exposed to all kind of injuries and chemical, mechanical, and so they need to be very often replaced. So these tissues keep a very high capacity to divide. However, other more specialized cells lose this capacity. For example, muscle cells, once you reach a certain number of muscle cells, they stop dividing, and they have a very low capacity to divide. Neurons, the brain cells, completely lose the capacity to divide. Once they're fully specialized, they'll lose the capacity to divide. And that's the problem. Why? Because you know when there is a spinal cord injury or brain injury, those cells cannot be replaced. Now, we are trying to overcome that as well, but it's very, very hard because they lose that capacity. So, what is actually happening? Why some cells can and some cells cannot divide once we reach the full growth and development? All cells are part of something that's called a cell cycle. That cell cycle has different phases. It's called G1, S, G2, and M phase. During the G1 phase, the cells just do what they need to do, right? They're fulfilling their normal functions. And during that interphase, at a certain time point, the signals arrive, and decision is made whether the cell needs to get into the division or not. If the signal is such that the cell needs to divide, then it actually enters in the next phase. It's called the S phase, and it stands from the synthesis, because the only thing that's happening in this phase is, and the most important part, of course, is that DNA is replicating. The DNA that's going to divide into two new cells. During G2 phase, another interphase, what's happening is cell is getting ready and preparing to divide, forming special proteins that are important for the cell division. Also, the DNA is forming the chromosomes, and they're ready to divide and split into two new cells. The cell division itself, or the M phase, or mitosis, is actually the shortest period during the cell cycle. And as you probably know from your high school, it's divided into different subphases. Prophase, metaphase, anaphase, and telophase, where actually chromosomes are lining up to correctly divide to two new cells, and then we have the division of the cytoplasm and formation of two new daughter cells. So this is a cell cycle. So what is happening? Cell, as needed, is going through this cycle over and over again, unless there is no need for cell to divide. If the cell doesn't need to divide, if there is no need, if we reach a certain growth and development, and there is no need for the tissue renewal, the cell actually exits this cycle and goes into so-called G0 phase, where it just does what it needs to do, but doesn't bother to even get into the new cell division. So it depends on the type of a cell, as I told you, whether it's an epithelial cell or a neuron or a muscle cell. And this cell cycle is differentiated, it's actually regulated at the molecular level. So there are different checkpoints here. This one is the most important one. It's called G1 checkpoint. Then we have, and then actually, of course, why is it most important? Because if there is no need for a cell to divide, why would it replicate the DNA? But why do we have then more than one checkpoint? Because even if the cell passes this initial S phase and enters the G2 phase, something can go wrong, okay? And so there are a couple of other checkpoints where the system is regulating whether everything is done properly and still deciding whether the cell is going to go into the full division or things are going to fall apart and be aborted. So it's working something like your dishwasher or a washing machine. You know, you have this control button where, you know, at a certain, you program your dishwasher for like a hot or warm, and at certain places it stops and decides whether it needs to do washing or it needs to put the hot water or it needs to put the cold water. So it's a very similar system. So at this checkpoint, the cell is actually looking at all the control mechanisms at the molecular level, and it either continues on in the S phase or just stops and exits into the dormant state. Different signals are very important for the control of the cell cycle. Some of them are internal at the molecular level, and some are external signals that depend on the amount of nutrients, growth factors, and also some mechanical responses. So we'll discuss a couple of important external stimuli that are important, or factors that are important for regulation of the cell division. One is the density-dependent inhibition, and the other is the anchorage dependence. In the normal situation, and how do we know this? Of course, most of the things that we learned about the cells are coming from in vitro studies. So if you take cells and you put them in a dish as a cell culture, what is happening? The cells are anchoring themselves to the bottom and dividing until when? Until they cover completely the bottom of that glass dish. But what is happening then? They stop dividing. That's called the density-dependent inhibition. Once the normal cell reaches that full cover of the dish, it stops dividing. Also, there is anchorage dependence, which means that if it doesn't have a substrate, a surface to attach to, it's going to stop dividing. And this is the main difference between the normal mammalian cells and the cancer cells. In the case of cancer cells, we lose these two external signals completely. Cancer cells do not respond to density-dependent inhibition. They keep dividing. And also they don't respond to the anchorage dependence. That means that they can divide endlessly, forming the clumps. And thus they are endangering other cells and other tissues from the point of getting the nutrients, getting the oxygen, and removing the waste. So once you have, and so there is a fine line how you define the cancer cell. At the beginning, what biologists like to do is not to define this kind of cell as a cancer cell, but they talk more about the transformed cell. So at the beginning, you have a cell that's transformed in such a way that actually it's not exhibiting this inhibition of cell division. And they tend to divide and keep dividing. But once they form a certain growth, then we call them tumor. And we'll see then how it becomes the cancer actually. So what would be the properties of these transformed cells? They have unusual number of chromosomes. Why? Because they keep dividing so quickly, and they don't care about any control mechanisms, that very often you see the cells with unusual, abnormal number of chromosomes, because the chromosomes don't divide between new cells equally. Then they have abnormal metabolism, because the only thing they care about is producing enough energy for dividing. They lose normal cellular functions, they lose the density-dependent inhibition, and they lose the anchorage dependence. So these are some of the easily recognized stages of the tumor and then later cancer development. So we start from the normal cell, and I can tell you right now, so what do you think 90% of the cancers are coming from? From what tissue? Epithelial tissues, not only skin, any epithelial tissue. So it can actually be found, cancer cells can be found in any organ, right? But the primary sites of the cancer are any types of epithelial tissues that you can find. But those epithelial tissue can be lining your lungs, can be lining your kidneys, can be lining your stomach. But why? Because they have the highest potential of dividing. So we'll see, of course, that the secondary sites can be any other tissue, but that's a different story. So the primary site of 90 to 95% of the cancers are different epithelial tissues that are found in your body. So at the beginning, what's happening, we have a stage that's called metaplasia, because cells just lose their normal morphology, lose their normal number of chromosomes, lose their normal functions, but still we're talking about transformed cells only. Then we have a situation where what we call a dysplasia. And in the talk that's coming after mine, you'll hear from a clinical pathologist more about how morphologically these different stages look. So there is a loss of cell uniformity, there is diversity of nuclear size and shape, and of course the first thing that you see is that there are more cells in mitosis. There are a lot of cells in mitosis. That indicates that actually it's happening more frequently. And this is called actually pre-malignant change or carcinoma in situ, okay? We still don't kind of call it a cancer. And there are fine lines, I know, and different people are calling it differently, but we still don't talk about this as a cancer. Now, if there is a growth or a new growth and a site, then we talk about neoplasia. Neoplasia means the new growth, and that is actually the tumor, okay? When you have something that's out of the boundaries of the normal tissue. And these are some examples of malignant epithelial skin cells, and you can see that a lot of cells are in the different stages of mitosis or the cell division. And here too, they are fast growing and they're in different stages of mitosis. And another thing that you can notice is also that the ratio, and that's important for some of optical modalities, that the ratio of nucleus and cytoplasm is changing because of these very frequent cell divisions. There is less and less cytoplasm because cell doesn't have time to replenish all the cytoplasm. So a nucleus versus cytoplasm ratio is increasing. So we can say that a cancer cell is nothing else but a pathological proliferation of your own cells, in most cases epithelial cells, that completely lost the control over the cell division. But how does it start? Why does it start? What is actually happening? And I also here wanted to differentiate between two types of cancer or tumors. One is benign, and those are the tumors that are slow growing, that are capsulated, that stay where they are, they're non-invasive, and they do not metastasize. On the other hand, this is what we are scared of, and those are malignant tumors. Those are the ones that are fast growing, they don't have capsule, and we'll see that the main problem with these, and those are the ones that are actually fatal, is that they are invasive from the point of releasing the cells, that are able to move around the body and start the cancer or the tumors and the secondary sites. So the most famous paper that actually did the review, and I recommend this as a reading if you're ever dealing with the cancer, is this review paper by Hanna and Weinberg in 2000, and it's called The Hallmarks of Cancer, that explains and describes the main properties of the cancer cell. It's probably the most referenced paper ever in the science literature. I think this is the old number that I have, 10,000 citations. It's probably more than that. 10 years later, as we are accumulating new knowledge about the cancer cells, they released another paper that's called The Hallmarks of Cancer, the Next Generation. And that gives new properties of the cancer cell that we need to take into account in order to better understand how the cancer cells develop. So I would like to go briefly over these main properties of the cancer cells. So there are, according to the first paper from 2000, what was that? 2000? 2000, yes. There are 6 essential traits shared by most, if not all, human tumors. And those are self-sufficiency in growth signal, insensitivity to anti-growth signal, evasion of apoptosis, so avoiding apoptosis, ability to replicate endlessly. So we know that these are so-called immortal cell lines, sustained angiogenesis, and the tissue invasion and metastasis. So you can see that not only did they change, so they just divide, and you think they go crazy, they are actually very smart, and they can often outsmart not only our immune system, but a lot of control mechanisms in our body. So what about the self-sufficiency in the growth signal? Well, in the case of normal cells, we mentioned that there is a need for a lot of signals to come to the cell in order for cell to decide to divide. So normal cells do not proliferate without such a signal, and they are usually extracellular, and they are transmitted through the membrane, through the receptors, and then by binding to the receptors on the membrane. Well, tumor cells don't need these signals. They couldn't care less if they're present or not. They are either have ability to mimic those growth signals, or they just produce them themselves. And thus they are able to stimulate themselves into the proliferation. So how is this happening? There are different ways, and that's the problem. The problem with the cancer that we have, the most important problem that we have with the cancer, is there are different reasons and different pathways the cancer cell can develop. So you have to target different things in different cases, and that's the main problem. So in a normal cell, there are certain genes that are called proto-oncogenes. Those proto-oncogenes are the genes that regulate the cell replication and differentiation in the presence of those mitogenic stimuli. But guess what? If those change, if those mutate or change in any way genetically, they turn into what we call oncogenes. So oncogenes are actually our genes that are changing in such a way that they don't need these external stimuli in order to cause the cell division. So they promote the cell growth and division in the absence of mitogenic stimuli. So how is that happening? Different reasons, different ways. Yes, there are viral oncogenes. Yes, there are some viral cancers. How is that happening? Well, if a virus, and there are about 15% of the cancers that are viral in origin, and if the viral oncogenes insert themselves into our DNA, they can actually cause the change in the control of genes that are in charge of controlling the cell cycle. But very few actually viral oncogenes are causes of cancer. in the case of non-viral oncogenes, we have mutation, translocation, insertion, amplification, and all these different genetic changes can cause the change or mutation of proto-oncogenes into oncogenes that produce oncoproteins and then cause cancer. Yes?
So I can understand it's viral oncogene. So you're saying if it's viral, could it translate from person to person, or is it?
No, it just inserts itself into our DNA and transforms our cells.
Okay.
But it's possibility. There are certain, if that virus resides, like papillomavirus, it could be transferred. Yes?
Whatever I know about cancer, it takes time to mutate.
Oh, yes.
Years.
Yeah.
So
W-
that's true in case of virus mutation also?
Yes, it takes, yes.
In 1 year, or it also takes time?
It depends. Because it depends also on, we'll see in a moment that it not only depends, it depends where it's inserted, right? Is that a cell, I don't want to scare you, but I always tell my students, there is a lot of cells in your body that are transforming all the time, but your body just fights them off. So they are either destroyed through apoptosis or by your immune system. So the question is, what is the condition of your body in general? Is your immune system strong? Are you taking care of yourself? And are there other mutations that are stimulating or inhibiting the gene expression that's oncogene? Because we'll see that it's not enough to have one mutation, one change, but accumulation of mutations. And that's why you have to also know your genetic history of your family. If somebody already carries a gene that's a predisposition for a certain cancer, then you're adding more mutation to that one is what we call genetic burden. If you don't have the genetic mutation, it takes many more mutations in order for cancer to develop. But it's also the exposing yourself to all kinds of cancerogens, like we know the certain compounds, radiation can also cause the damage that can stimulate the mutation. So, but let me show you how is that happening, okay? How is that self-sufficiency in the growth signal actually happening? So this is a proto-oncogene. It's one gene in a DNA that's actually regulating some part of the cell cycle, okay? So what can happen? One example is that it can be a translocation where gene is moved to a new position, a new site, where it's under the new control. So instead of like having one normal growth signal, you have the overexpression of that protein, and suddenly cell thinks that it needs to divide more than normal. Gene amplification, if this gene is amplified, if the multiple copies of the gene by mistake are made in a cell, again you have overproduction of that oncoprotein, and that can cause the stimulation for cell division. Point mutation in a control element, in a promoter, can cause the overexpression or overproduction of a protein, and thus normal growth stimulating protein production of in excess. Point mutation within the gene can cause hyperactive or protein that's resistant to degradation. So all these examples are showing that different things can cause the transformation of one gene, a proto-oncogene, into oncogene, and thus cause the transformation of the whole cell. But we are far away from cancer yet. Okay, so one thing that you need is self-sufficiency in the growth in the growth signal. So this cell, for some reason, for some genetic reason, doesn't need the growth signals from outside. It produces its own. But at the same time, there are also anti-growth signals. There are signals from outside the cell that are actually inhibiting the unnecessary growth and division of the cell. And they're also found in outside extracellular matrix, and they block proliferation. And most of them actually, most of those signals, proteins, are actually pushing the cell out of the normal cell cycle into the G0 phase. However, cell has to be responsive to those signals, but in the case of the transformed cells, that response is lost. So cancer or transformed cell can actually completely ignore these signals and avoid them. So, and they also can avoid the terminal differentiation. So once they start dividing, they don't differentiate. And because they don't completely differentiate, they keep the ability to divide. So this is usually, you have to make sure that you understand the complete picture. It's never just one thing that causes a transformed cell. That's a combination of different things. For example, so one thing is, this is the cycle, the pathway that shows you how the protein stimulates its own cell cycle. So there is a growth factor that's attaching to the membrane receptor, and that activates the very important so-called RAS protein, G protein, that through the chain reaction actually causes the gene expression and stimulates the production of oncoproteins. Well, this doesn't exist in the transformed cell. This is not needed. So the cell does not need the growth factors from outside, and they can do this part by themselves. At the same time, what's happening, so this is the first part that we had on a previous slide. At the same time, there is an active form of a protein that's called P53. And this protein is called guardian angel of the cell, because in most cases, this protein can actually inhibit the transformed cell to divide. It's the main anti-growth signal. However, in most cancers that we know of, there is a problem with P53. So if P53 suffers a mutation, if it suffers a change, there is no guardian angel, and there is no anti-growth signal, and the transformed cell can keep dividing. So in most cases, as a summary of both, if you have the cell that's transformed, you usually have both. You have cell cycle that's overstimulated, and then it's not inhibited. And that causes the over division or overproduction of the cells. And this is what I wanted to show you. What the transformed cell is, the reason for the transformed cells, the cause of the transformed cell, is actually the progressive non-lethal DNA damage leading to uncontrolled cell divisions. So first of all, you have to understand that, and the fundamental change that's happening in a transformed cell is the DNA damage, but the one that's not lethal. If the DNA change is lethal, the cell dies, right? So you have to have a DNA change that's going to affect the cell cycle, that's going to affect the cell division, and it's not going to kill the cell at the same time. And also, it's not enough to have only one change, but usually you have the accumulation. This is the example of the multi-step model for the development of colorectal cancer. Usually one change, mutation in the DNA is causing formation of maybe some small benign growth like a polyp. This is why it's important to remove them, because if there is more changes, more mutations, that can develop, especially the loss of the tumor suppressor gene P53 normal function, that can cause the growth of the tumor or development of the cancer. For the next essential trait, we need to differentiate between the apoptosis and necrosis. Both are actually indicating the cell death, but in a very different way. We didn't know about apoptosis until about, I don't know, 15, 20 years ago, because we thought, well, when the cell dies, it dies, right? But there is a very big difference between the accidental cell death when the cell is damaged, and when we are talking about pathological process, when the cell swells and bursts and then you have inflammation or infection and macrophages in your immune system are cleaning up the site, versus the apoptosis, which is actually a cell suicide or programmed cell death. Your body reacts to any change in normal cell function in a way that actually cell recognizes that it should be destroyed, and then in a very organized and synchronized way, cell and nucleus are being degraded step by step inside the cell membrane. So there is no exposure of the cytoplasm and the enzymes inside the cell until they're all packed into little vesicles safely, and then they are actually packaged into little vesicles, and they're detaching from the cell, and then they are destroyed by macrophages. So in this case, there is no inflammatory reaction, and it's a very organized process. And in a lot of cases, this actually prevents the formation of tumors. So if there is, in many cases, if the transformed cell identifies itself as a transformed cell, it actually starts this apoptosis, this programmed suicide. But of course, in addition to removal of transformed cells, it's a very important, we now know that this process is very important in the growth and embryonic development, because it's actually shaping our body during embryonic development. So the tumor cells, the expansion of this tumor cell population depends on the rate of proliferation, but also on the rate of their how quickly they're destroyed. So if our body is able to destroy these cells quickly, faster than they can divide, we are okay. However, a lot of these cells, transformed cells, have ability to avoid the cell suicide, which is the major barrier for cancer. The second barrier, of course, is your immune system. So there are very defined steps of apoptosis. Cell is usually destroyed within 30 to 120 minutes. Cellular membranes are disrupted. The nuclear skeleton is broken down, and the chromosomes are degraded. Organelles and nucleus are fragmented. They're packed, and it's called the pycnotic nucleus. They're packed into the vesicles, and then they leave the cell within in a form of a small vesicles. So there is no inflammation on the site. So in the case of normal cells, we know that the normal cells have only a certain number of divisions. So your cells don't live indefinitely. We know that they have ability to divide only a certain number of times. So if they become damaged or too old, then usually this p53 protein starts the apoptotic process, and these cells are destroyed. However, the tumor cells have the ability to avoid this apoptosis, and as I told you, about 50% of human cancers show the loss of the 53 protein malfunction. That protein is not starting the apoptotic process, and the damaged DNA is transferred to the new cells. Another thing that's not very good from the point of transformed cells is the ability of these transformed cells to divide endlessly. Very often, when we culture these cells in a dish, we talk about immortal cell lines. And you probably heard about HeLa cells, right? Those cells were isolated from female ovarian cancer in the 50s, and that cell line is still alive and well. However, if you try to culture a normal mammalian cells, they have a certain potential to divide, and usually that's about 60 to 70 divisions, and then they stop dividing. So there is a certain replicative potential. And this is the reason behind that is that with every cell division, there is a loss of a piece of DNA from the end. So once that reaches a certain critical point, that's called telomeric DNA. So when that DNA reaches a certain critical point, the DNA cannot replicate anymore and the cell cannot divide. Yes?
So why does that loss happen?
What is, do you know how the replication of DNA takes place? Roughly, yeah. That's all in the mechanism. So you have two strands of DNA, right? And one strand of DNA is duplicating in a normal way, right? And the other, because how the DNA polymerase is working, it has to work in backward sequences. You know that? Okay. So there is always a part of DNA that cannot be replicated because it has to attach to the part of DNA in order to, so you have a strand of DNA, and then you can't replicate this way, you have to replicate this way, and the pieces of DNA that are formed are connected together with a ligase. Okay, and there's always a piece of DNA that cannot be replicated because the DNA polymerase has to be attached to something. So when it attaches to the old strand of DNA, there is the part of the new DNA that's missing. So there is always about 50 to 100 base pairs that are not replicated. So in our DNA, at the end, there is always the, well, you can call it a dumb part of a DNA that doesn't really code for anything because it's there just for the replication. That part, that's the only use of that part of the DNA. But with every DNA replication and the cell division, you lose 50 or 100 base pairs. And when you reach the critical point, when you start getting to the genes that are actually expressed, it stops and it can't be replicated. Yes?
How do HeLa cells do that?
All cancer cells, they have a special enzyme that's called telomerase enzyme that actually replaces those lost base pairs. And I'm telling you, that's one reason why actually cancer cells are extremely, I don't want to say, of course, smart because they don't have brain to think, but they're very adaptable. They can adapt to anything you throw at them. They find a way to work around it. So what they have, they upregulate the enzyme that's called telomerase enzyme. That's the enzyme that actually can replace those lost base pairs of DNA, so that actually that enzyme adds more repeating, totally nonsense base pairs, but you don't lose actually the length of DNA because of that. And that's why, and that permits the unlimited multiplication, and that causes these cell lines to be immortal. And most of the cancer cell lines are immortal.
So the normal cells only have 60 to 70 divisions. So how many days does these divisions correspond to? Like, how long?
You mean in the body or in the culture, or?
During the cultures.
During the culture, you have those passages, right? So it depends a lot on the type of the cell, but they can't even go through the 60 or 70 passages. It really depends on some cell lines go through, I don't know, 5, 10. If you go to 15 or 20, you're already having bad cells. I don't have much experience with a lot of cell lines. I'm working with some, so I don't know exactly, but you better freeze them down. So you pass them a couple of times, and then you just freeze them, and then when you need them, you take them. But it really depends on the type of the cell line. It again depends on stem cells and young cells have a higher ability to do that than the older cells. It depends again on a type of the cell.
Can we turn that up and get a little bit more mileage out of our cells? Or?
That would be good, I guess. But the whole problem with getting old, or our cells getting old, is everything is in balance between making and breaking. DNA is accumulating a lot of genetic changes. There are a lot of control mechanisms, but it's a balance. Can you keep up with making and repairing everything that's breaking? And as we are getting older, or our cells are getting older, I don't mean, I mean cells getting older from the point also of days and months, not only years, can those cells keep up with all these changes, or they give up and die? Control mechanisms just can't keep up with all the changes that are happening. It's possible. It's possible, yes.
So there's an internal upper limit for cell division. Is there differences between different size animals with that number of divisions going to be? For example, with a large animal like an elephant, primary cell division lasts longer than very small animals.
I never looked into that. That's an interesting question, but I don't think so, because the size of the cell is pretty much the same. Whether you're talking elephant, human, they're all around, what? You tell me. 10, 5, 10, 15 microns. So the size of the cell is pretty much the same. So I don't think so. There are a lot of speculations, why are some animals living longer than the others? And I think it has to do sometimes to do something with the size, but it's not from the point of cell replication. It's from the point of metabolic rate and the ratio of surface and volume. That's a totally different physiological problem. It seems that the small animals have a much higher surface to volume ratio. So in the small animals, everything, all the functions have to be more intense, like breathing, eating, more oxygen, and so on. They use themselves up much faster than the big animals. But I think if you isolate the cells themselves and you put them in a culture, I think that you don't see much difference. It's more at the level of the organism itself. Okay, so the next big thing about cancer cells is sustained angiogenesis. In the case of normal cells, all normal cells, need the blood supply for the nutrients, oxygen, and to remove the waste and carbon dioxide. So all cells in your body are, you can say about 2 to 3 cells at the most away from the blood vessel or the capillary. Okay, can't be any further than that. So we depend actually on the presence of the blood capillaries and the blood flow. So angiogenesis, or the blood vessel growth, is a very important part of normal function of all our cells. Now, in the case of the tumor cells, the tumor is growing, of course, and forming the new growth. And you would say, well, how are they supplied with the nutrients and the oxygen? Well, they have ability to develop their own angiogenic stimuli. So they have ability to activate angiogenesis. So in most tumors, you see the new growth of blood vessels. And that's one of the things that very often is the target for imaging, and also target for treatment. Not to target the tumor itself, but to target actually the growth of the growth of new blood vessels. Very often, if the tumor is growing too fast, you see the necrosis within the tumor, inside the tumor, meaning that the cells inside the tumor are so deprived of the nutrients and the oxygen that they start dying, but tumor couldn't care less. The outer cells are still dividing very extensively, and they are supplied with enough nutrients, so they keep on living and dividing. But in a lot of cases, they have this angiogenic switch, and they start their own blood vessel growth, which, as is mentioned here, offers a very important therapeutic target. And of course, this is probably the most important hallmark of cancer, and that is the tissue invasion and metastasis. And that is the ability of cancer cells to leave the primary tumor and to colonize other parts of the body. And this is what's actually lethal. 90% of lethal cancers or cancer deaths are due to metastasis. As long as the tumor is in a place, it's rather safe. Once it starts leaving, so what is actually happening? Cells have receptors and adhesion proteins that keep them together in the case of normal tissues. And there are certain proteins that are called integrins that link the cells also to extracellular matrix on ECM. And that's why our cells pretty much stay where they are. Your epithelial cells stay where they are, your kidney cells stay where they are, and so on. However, in the case of cancer cells, that adhesion and those links are lost. In the case of invasive cancers, these cells just separate from the primary tumors. They actually, and this is the case of the breast tumor, they leave the primary site. They actually go into, most often into the bloodstream, although they can also be transported by the lymphatic system. And they adhere to the basement membrane of the blood vessels. They're able to leave the blood vessels by extravasation and form micrometastases, or what we call the micromets. And in some cases, those micromets can develop into macrometastases and even cause the angiogenesis or the growth of the new blood vessels. It takes time, sometimes years, for this to happen. And sometimes the tumors just stay where they are, and sometimes they are able to invade other tissues very rapidly. It all depends on the type of the cancer cell and the type of the change that occurred in the transformed cell. So what are the main properties of the cancer? It's a multi-step process that's starting at the genetic level, but also it's recognized as a phenotype, at the level of the phenotype. So the fundamental thing is that it's a genetic change that's non-lethal, that doesn't kill the cell. In most cases, we have a clonal proliferation of only one cell. So most cancers are monoclonal. So just one, sometimes it's only one cell enough to change in order to cause the cancer. And it's not destroyed, of course, by your immune system or apoptosis. And ability to migrate and invade other tissues. Now, I would like to go very quickly. It's already my time out, but very quickly just to point to you that there are new findings in that 2011 paper. There are new findings that seem to be very important for cancer and other capabilities and properties of cancer that are very important for us to know about in order to target the cancer. Those are two emerging hallmark capabilities, two enabling characteristics, and the significance of tumor microenvironment. We cannot emphasize anymore the importance of microenvironment, because we now know that there is a very important interaction between the transformed cells and the microenvironment that's either going to stimulate or inhibit the further growth. So those are avoiding immune destruction, deregulating cellular energy processes, genome instability and mutation, and tumor promoting and inflammation, things that we didn't know about 10 years ago. So in the case of genome instability and mutation, we know that the normal cells have ability to monitor the DNA replication and the changes, and if needed, use their repair systems to repair the mutations. But in the case of the transformed cells, especially in the case of p53 protein, those repair systems are not functioning well. And that's the main reason why these transformed cells can survive the destruction. For a long time, we thought that inflammation and the response of our immune system is the main important thing in fight against cancer in our body. But now we know that there are certain immune cells that not only not actually attack the tumor, the cancer cells, but actually support the cancerogenesis. And this was really the kind of a very sad kind of discovery, that there are certain inflammatory cells that, especially during the early stages of cancerogenesis, that can release mutagens and accelerate the further development of malignancy. So basically, in a way, our body is turning against ourselves. Reprogramming the energy metabolism or metabolic switch. You know that our cells depend a lot on the oxygen. Why? Because they use the oxygen to transform the glucose into the pyruvate and the CO2. But in the case of tumor cells, they're so piled up that very often oxygen cannot penetrate to all the cells if the angiogenesis is not developed enough. But guess what? Some of the cancer cells actually switch completely to anaerobic metabolism, to glycolysis. So they can drive their glucose degradation only to lactate under hypoxic conditions. And very often within the tumor, you can find the mixed population of both lactate producing and lactate utilizing cells. So they kind of help each other. One population of cells uses very quickly glucose to the level of lactate without the oxygen to produce energy that's important for the cell division, while the other cells that are maybe more exposed to oxygen can use that lactate to completely degrade into the CO2 and water and further produce ATP. So you can see that there is a lot of adaptation within those cancer cells to kind of find the way around some conditions. Immune system, in a lot of cases, of course, our immune system is able to respond to the presence of transformed cells and destroy them, whether those are macrophages or lymphocytes. But we now know that a lot of cancer cells have ability to avoid the immunological destruction by enabling disabling those cells within the immune system. Tumor environment. That's something that we now know a lot about, and that is that within the tumor, we don't have only the cancer cells that are just dividing. That environment is much more complex than we thought before. There are other cells, like the stromal cells and the extracellular matrix, that are very important in the further development. So this was our previous knowledge, or how we imagined the tumor. Now we know that there are other cells that are a very important part of the function of those transformed cells, and they're pretty much recruiting the other cells in our body, especially stromal cells and immune cells, to work for them. And that's what's kind of scary. And so that dynamic interaction between the cancer and stromal cells actually leads to the multi-step transformation of normal cells into malignant cells. So there is a very strong, and we now know that also even the density of the extracellular matrix is very important for the further development of cancer. We don't even know yet, but there is a very strong interaction We still don't know whether the cancer cells produce more collagen to make the extracellular matrix stiffer, which promotes the development and the migration of cancer cells, or the stiffer environment actually promotes the cancer. But we actually believe that there is a mutual interaction between the cancer cells and ECM. So therapeutic targeting, now that we know more and we are discovering every day new hallmarks of the cancer, that enables us to target some of those characteristics and properties. So more effective, we now know that more effective therapies require actually co-targeting. We cannot only target one of these things, the angiogenesis or ECM. We need to do co-targeting, and we need to, at the same time, look at the multiple capabilities and enabling characteristics in order to overcome most of the cancers. But you can see that there are so many different facets of the cancer that we need still to learn about in order to effectively fight cancer. And I'm sure if you read those two articles that you'll find much more that I had to skip over. Do you have any questions? Yes.
Yeah, you mentioned that the nucleus to cytoplasm ratio increases in cancer.
Yes.
But also, don't cancer cells just have larger nuclei in general?
Well, I don't know if they have the larger nuclei or it appears larger because it's in cell division. So you have the mitotic spindle, you have chromosomes, but I don't think it's much larger. I just think it changes its morphology completely. But I think what's really decreasing is the amount of the cytoplasm, because after normal cell division, there is a period of interphase where, because you divide cytoplasm into two, right? And so the daughter cells are receiving half of the cytoplasm of the mother cell. And there is a period during the interphase where they replenish the rest of the cytoplasm, build their own organelles, and so on. Well, that phase is cut short because it goes into the next cell division.
Yeah, because I know sometimes in pathology you look at nuclear size to determine.
Right.
So, but you're saying maybe it's not actually.
Dr. Tangela, who is going to give a next lecture, is going to probably answer that question much better than I do. Dr. Tangela?
What's the question?
The question is, in transformed cells or cancer cells, is the nucleus really bigger, or the ratio of nucleus and cytoplasm is changing?
All of it.
All the above.
Yeah.
Why is that? Obviously, the nucleus-cytoplasm ratio is changing because the cytoplasm doesn't have time to...
It depends on the tumor, actually.
It depends on the tumor.
Yes.
Right.
Some tumors have a very high nuclear to cytoplasmic ratio, where the nucleus is pretty much the entire, tumor cell. We have a very thin rim of cytoplasm. Example being a small cell carcinoma of lung.
Yeah.
So pretty much everything is a nucleus and very thin. Whereas squamous cell carcinoma, which are well differentiated, have abundant amount of cytoplasm. So the cytoplasm to nuclear ratio or nucleomegaly or lack of cytoplasm, all these factors don't per se define the malignant cells. All depends on what kind of tumor biology exists because of genetic defects.
Okay, so that's not a good marker to differentiate tumor or not.
In generality,
Okay.
you can say that.
Okay.
But specifically, yes. Now, individually, all the factors you say make sense.
And why would be the nucleus bigger than normal?
Why is the nucleus bigger than?
Normal, in some cases.
Oh, because it's, again, it all depends on genetics, okay? Generally, a tumor cell is a fast-growing tumor cells have a lot of chromosomes that have actually unfurled, and they are all like kind of loose. Whereas in a normal cheek human cell, the nucleus in a cheek squamous cell is so thin and hypnotic because everything is compact, and the type of genetic material that's required for the cell functioning is functioning. Now, if the tumor has got malignant and is rapidly growing, you can imagine how much of ribosomes it has to make, how much of transport, tRNAs it has to make, how much of DNA it has to replicate. So the nucleus obviously starts unfolding. This is specifically true in a lot of cancers, including kidney cancers, where we grade the tumor based upon the largeness of the nuclear size. We call Perman grade. So low-grade renal cell carcinomas have a smaller nuclear size than a high-grade renal cell carcinomas, which have larger nuclear size. Make sense?
Wouldn't we reach a limit at some point in the sense that if the cancer cell is going to keep dividing, my understanding is that I don't know anything about biology, but using some of the materials from the cytoplasm in conjunction with the...
Absolutely.
So
not only then using the material from the cytoplasm, but also using materials from outside.
So basically my question is, doesn't this actually help to inhibit or slow down the division of the cancer cells?
Oh, it does. That's why the tumors have tumor necrosis. Squamous cell carcinomas and high-grade tumors, leiomyosarcoma, has rapidly proliferating tumor cells, and then smack in the middle, it has like an area of necrosis because the tumor is dividing so fast that it can barely feed itself. Right? So you're right.
So there are some, I try to present some general things about the cancer cell, but there are a lot of exceptions. And every transformed cell is a little different. Every cancer is different and depends on conditions and circumstances and type of genetic change that occurs. So, yes.
So the telomerase enzyme that is helping at the base pairs. So I'm guessing the gene is present in the normal cells, just it's not expressed.
It's not as active, yes.
Okay.
In normal cells, it's present.
It's present. So what
Yes.
kind of function it has in the normal cell?
It does the same thing, but I think the telomerase enzyme is actually more active in a young, fast developing, during embryonic development. And then once we reach the full growth, we have a lot of enzymes that are suppressed, but that are left over from our ancestors, right? So a lot of enzymes are just either completely suppressed, a lot of proteins are either completely suppressed or they're suppressed during the development. And I think the telomerase is one of those examples when we reach a certain growth and development that they just cease to be active. Any other questions? I guess we can have a few minutes break, and then Dr. Tangela is gonna. Hi, how are you? She graduated yesterday.
Oh my goodness! What is
Yes.
she going to do?
She doesn't know yet. She just graduated from architecture.
Okay.
So